Okay, so the company was founded 6 years ago when Karen was tasked with the concept of marrying engineering principles with biotechnology to develop a 3D human trabecular meshwork model ex vivo that could test compounds and correlate to data seen in the clinic. And so being tasked with that, she worked very hard using her engineering background to pull that together. And today we're servicing many clients within the ophthalmology space on their drug discovery, testing needs from beginning of the process all the way down through the clinical side.
And really our mission and what we really do at Glauconix Biosciences is trying to develop tools that is going to allow pharmaceutical companies, new biotech, early stage companies come up with the best and most innovative glaucoma drugs, and ophthalmology in general, because we're also working in other areas of the eye, not just glaucoma, looking at other indications as well, and really just making sure that we're providing models that are human-based, that are really mimicking not only the biology but also the physiology and the pathology, right? Because pathology, I think, when we look at the disease and how are we really going to create these novel treatments, the biology and again pathology of a disease is extremely important, and we want to be able to mimic that as well. And you know, glaucoma is not just one. You don't say, oh, I have glaucoma and this is what I'm going to treat, right? There are different types of glaucomas, and you really need to make sure that, you know, we can serve also those different types. You know, what kind of medications, what kind of devices can we serve for very specific types of glaucoma? And the same thing happens for other diseases more towards the back of the eye. So I think that's something that we are committed to be able to provide these tools that are, again, more biology and pathological and physiologically relevant to in vivo or human settings.
So we currently have a few different models to support glaucoma, one being through the trabecular meshwork, our HDM model, and then the other being a co-culture model that then incorporates Schlemm's canal. Beyond that, as Karen mentioned, we are going to be looking at developing some models to support back of the eye areas as diabetic retinopathy and macular edema. So we're working on that in 2020 with the hopes of coming out with a model in this year.
But we're also constantly looking for, you know, what are the unmet needs in ophthalmology in general, to be able to provide, again, the highest quality of science and tools that are really going to help move compounds and therapies faster from proof of concept all the way to clinical trials and marketing.